All right, thank you very much, Kelvin, for that introduction, and thank you everyone for being here. So today in my talk, I'm going to address this question: does the IL-13 system contribute to Parkinson's disease? Before I address this question, I'd like to talk about my journey, my trajectory into research science. And if I'm going to start with my journey into research science, then we should start with this question: to cook, to fly, or to research? So why to cook or to fly? Well, let me tell you that we need to go back to high school. So during high school, I knew that I wanted to either be a chef or a pilot. So of course, the question now is, how in the world did I become a research scientist? You know, I asked myself that from time to time, and basically the story goes like this. So senior year of high school, you know, I'm getting ready for graduation, and I mentioned to one of my math teachers, I mentioned to him that, hey, you know, I think I'm going to join the Air Force because I want to learn how to fly planes and helicopters because I want to be a pilot. And he tells me, if that's what you want to do, that's great, but I have a piece of advice for you. He said, go to college and get a bachelor's degree, because with a degree, you could enter the military as an officer. So I thought that was great advice. I didn't know that. So I said, yeah, let me go get a bachelor's and enter the Air Force as an officer. And so what ended up happening is I went to UCI, so UC Irvine, and I went as a science major, so pharmaceutical science, pharmacology. And basically, yeah, in high school, I did like science. That's why I went as a science major. I liked science because it was hands-on. I liked the laboratory part of science. That continued during my undergraduate. So in my undergraduate, I was doing okay in the science lecture courses. But in the science laboratory courses, I was doing good. And so at the beginning of my second year at UCI, I was in organic chemistry laboratory. And it was a night class laboratory. So I don't know if you guys are taking night class laboratories. Everybody just wants to get out of there, rush out of there, finish the experiment, and get the heck out of there. Everyone's hungry too. So anyways, I was in that laboratory at night, and the TA comes up to me after a couple of weeks in that course, and she says, hey, I noticed that you're like one of the only ones who doesn't want to rush out of here. You're always smiling when you're working with the chemicals and everything. You enjoy this, right? And I told her, yeah, I do. Like, this is fun to me. So she advised me. I got another advice now. She said, why don't you get some undergraduate research experience to see if you want to do research as a career? And I said, again, I was like, oh, why not? That sounds like good advice. And so, so this time here at UCI, I emailed about like 5, 6 professors, and 2 of them emailed me back. And I ended up joining a laboratory that was conducting drug addiction research. I found that topic interesting at the time, so I joined a drug addiction research laboratory. And then after I joined it, this was like in the middle of my second year. After I joined it, I ran across one of my friends at UCI. And I informed him that I was doing undergrad research now. And he tells me, oh, really? Well, do you know that if you qualify as a minority underrepresented doing undergraduate research in science, UCI has programs that will pay your tuition and fees and give you a monthly allowance, a monthly stipend. Now, I thought my friend was completely crazy, high on drugs, or both. I was already a month or 2 into my research experience, so I was already researching for free. I was liking it, I was loving it. And so I thought to myself, why would anybody pay me if I'm already been doing it for free? So what happened was we ran across each other again because we were in the same club. So at club meetings, we ran across each other again. And he asked me a couple weeks later when we see each other again, hey, are you getting paid now? And I told him, no, I'm still not in those programs. And he says, why not? You could be getting paid now. So I thought, okay, maybe he's not crazy. Maybe these programs do exist. And so I went to the UCI BioSci website. And indeed, at UCI, there's outreach and minority science programs that if you qualify as an undergraduate minority underrepresented in research science, you can get your funds, you can get funds for tuition and fees, so those are paid, and you get about $1,000 a month. Now let me tell you, as an undergraduate getting $1,000 a month, I felt rich. So yeah, so I was in this program, so it's a long name, but MARC for short. It's funded by the NIH. You could, as an undergrad, you could be in there for a maximum of 2 years. So I was there as a 3rd and a 4th year in that program. And I was also in this program. So UCI also has this MERT program, also funded by the NIH. And basically, that's a summer program, 2.5 months. You get research training abroad. Of course, I took it. It's completely free. They pay for everything, for your travel. They give you a stipend to live wherever you go abroad. And at the time when I applied, it was either you could go to a university in Mexico to get research training, a university in Spain, or a university in London. So I'm a Mexican American, and I wanted to go to Spain. So I was like, you're telling me I'm going to get more research training abroad, and I get to have fun in Spain? I definitely wanted to go to Spain. At the time, it was about a cohort of 10 of us. So I was the only one who chose Spain. Everyone else chose London. And to me, that was perplexing. So anyways, they wanted to go to the summer to London. And for safety reasons, UCI told me, look, everyone's going to London. You're the only one who wants to go to Spain. We can't have you by yourself over there. So go to London. So I was kind of bummed out. I was like, man, I'm going to London. And but it ended up being a blessing for sure. So in London, that summer between my 3rd and 4th year is where I got exposed to Parkinson's disease research, and I fell in love with it. So over there, I mean, I wasn't taking any courses, so I was fully involved in just research. And basically, I read a lot about Parkinson's disease. I was in the lab a lot, and basically, I got exposed to what's happening biochemically during Parkinson's disease and the death of dopamine neurons. So that sparked my passion for Parkinson's disease, which is what I still do. So it definitely was a blessing for me that I ended up going to London. And so I returned, and then it's my 4th year. I like research science, and I applied to PhD programs. And so I think I applied to about 10 or 12. I got accepted to one at the University of Miami. And I didn't want to leave my mother or my sister alone here in California. So I said, let me get a master's. And hopefully with the master's, I'll be competitive for a PhD program in California. So I did just that. And I went to Cal State LA for a research master's. Again, fortunately, Cal State LA also has this MBRS Rise Master's to PhD program. Again, it pays for underrepresented students in research science, your tuitions, your fees, and you get a monthly stipend. So I was fortunate enough to get that bachelor's pretty much debt-free, and then a research master's debt-free. And so, of course, after I get my master's, I still loved research science. I still liked it. I had a passion for it. So I applied again to PhD programs, and obviously I got accepted to the Scripps research program, and now that's what I'm doing. So, however, now I like to think about it in retrospect. I like to think that research science has allowed me to get a bit of both the chef and the pilot experience. So those programs, since I've been an undergrad, they've been funding my travel. I went to London, I went all over Europe, to Puerto Rico, all over the nation as an undergrad, and it was completely free thanks to the funding from that program. I was presenting at conferences and things like that. And of course, when you travel, you eat world cuisine, so I think that's the chef part to it, or at least I like to think of it that way. Okay, so that wraps up my journey into research science, and so now I'm currently a PhD student at Scripps Research, and what I do is I investigate Parkinson's disease. And so it's the second most common neurodegenerative disorder. It affects 1% of the population over the age of 60. Here, this illustration, it pinpoints a region in the brain called the substantia nigra. And so that's mainly characterized in Parkinson's disease research, in which dopaminergic neurons are lost. Over here on the right, what happens is that in a healthy dopamine neuron, you have an adequate amount of release of dopamine depicted there as red dots. However, in a Parkinson's affected neuron, what happens is that dopamine release is greatly reduced, consequently leading to a reduction in dopamine signaling, and therefore you get symptoms of Parkinson's disease. And so what are some of the symptoms? Well, Parkinson's disease has both motor symptoms and non-motor symptoms. Some of the motor symptoms include resting tremor. It includes postural instability. If you push slightly a Parkinson's disease patient, they tend to fall over. and as well as rigidity. Some of the non-motor symptoms include a loss of sense of smell. That's one of the first things that when someone gets Parkinson's disease, they lose their sense of smell. Dementia and depression. And so here are some numbers in regards to the economic burden of Parkinson's disease. So these numbers are from 2013. So you can imagine that these numbers are now a bit higher. So to the American society, each year, the cost exceeds $14.4 billion. Treatment costs alone are about $8 billion a year, and the indirect costs exceed $6 billion a year. You hear a lot that the population, the aging population, is increasing and that humans are living longer. So these numbers are projected to double by 2040. Now, a question in regards to Parkinson's disease is, what causes the loss of dopaminergic neurons in Parkinson's disease? So this is due to mitochondrial dysfunction, oxidative stress, and protein misfolding. And monogenic forms, what's interesting is that mutations in genes account for approximately only 3% of all non-familial Parkinson's disease. So the vast majority of people who get Parkinson's disease, we don't know why. We don't know. It's unknown. And so that has led to this notion in the field. Researchers believe that Parkinson's disease is due to both genetic factors as well as environmental factors, an interaction of these factors. And lately in the field, recently, researchers have begun to believe that inflammation could contribute to Parkinson's disease. Inflammation could be an immunological response to an environmental factor, so therefore it could represent an environmental factor. And so here we have a depiction of what inflammation could be doing in regards to the death of dopamine neurons. If we start here at number 1 in the dark red arrow right here, during inflammation, you have the presence of reactive oxygen species and nitric oxides, which can then lead to the death of dopamine neurons. The death of dopamine neurons then will activate glial cells in the brain, and you could also get infiltrating T cells. When glial cells are activated, they can release pro-inflammatory cytokines that will then keep a pro-inflammatory environment in the brain. Thus, this cycle will just continue. Now, currently, we don't know if inflammation is a cause or a consequence of dopaminergic neuronal loss. And in this model, there's no evidence specificity for dopamine neurons. However, the work in our lab, which I will show in the upcoming slides, suggests that specificity could be attributed to this cytokine, the IL-13 cytokine. And this is because dopamine neurons express IL-13 receptor alpha 1, in which this ligand can bind to and act directly on dopamine neurons. And so one question we can ask in regards to inflammation in Parkinson's disease is, if inflammation contributes to neuronal loss in PD, can genes regulating immunity represent genetic factors for increased susceptibility to Parkinson's disease? So this is getting into the field of biomarkers for Parkinson's disease. And so we initially started to address this question by going to online databases and also the literature. And what we found was on chromosome X, there's this PARC12PD susceptibility region in which the IL-13 receptor alpha 1 gene lies within that. Then on chromosome 5, there's this 5q32PD susceptibility region, and adjacent to that region, we have the IL-4 and IL-13 genes. So from now on, in the rest of my talk, when I mention the IL-13 system, it could be any of these components here. So what happens is that IL-4 receptor alpha forms a heterodimer receptor complex with IL-13 receptor alpha 1. And this heterodimer receptor complex, the ligands IL-4 and IL-13 can bind to it. And upon binding, they can elicit signaling pathways, two of which are the JAK-STAT pathway, the PI3 kinase AKT-mTOR pathway. And so what do we know about IL-13? Well, what we know from it is mainly from the periphery in the body, from immunology. We know that IL-13 and IL-4 are cytokines that are necessary and sufficient for the development of allergic asthma. They activate various cells in the periphery to induce the contribution to the pathogenesis of allergic asthma. But this is the periphery of the body. Parkinson's disease is the brain, is a central nervous system. So what about in the brain? What do we know about the IL-13 system in the brain? Well, our lab, what we have done is we've started to map the expression of the IL-13 system in the brain. Here in A, first we see staining results for TH. TH stands for tyrosine hydroxylase, which is a marker for dopaminergic neurons. And what we see in these sagittal sections of mouse brains is we see staining for dopaminergic neurons in the olfactory bulb. Recall that in Parkinson's disease, a loss of sense of smell occurs. We see staining in the VTA substantia nigra pars compacta and the locus coeruleus. Here, zoomed in, you can definitely see the staining in the substantia nigra pars compacta. So that's for dopaminergic neurons. What about for IL-13 receptor alpha 1? Well, if we zoom in, we see staining also in the substantia nigra pars compacta as well as the VTA. So in the brain, IL-13 receptor alpha 1 is expressed in neurons that are lost in PD. What about the other part of the heterodimer receptor complex? Well, with these coronal sections, we did a similar staining, and we can see staining results as well in the substantia nigra pars compacta for IL-4 receptor alpha. Now, this is the receptor. What about the ligand that binds to this receptor? Well, we did some immunofluorescence experiments. I don't want to go through all these figures, but I want to highlight B. For B, we have IL-13 fluoresce in green, and IBA1 is a marker for microglia in red. So if they co-localize, they'll form the color yellow, and indeed we see co-localization of IL-13 on microglia. And when we quantitated this, microglia accounted for approximately 82% of central IL-13. So the receptors are there and the ligand is there in the brain. So now that we knew that, we then went on to conduct some experiments. And we started with in vivo experiments in mice. And in these mice, what we did is we generated a group of mice that don't express the IL-13 receptor alpha 1 receptor. So they lack the receptor. And then we subjected these mice to a substance called LPS injections that will then induce dopaminergic neuron loss. And the question we wanted to ask was, if we take away the receptor from mice, will it have a protective effect or augment the dopaminergic neuronal loss? And what we see is that when wild-type regular mice and the mice that lack the receptor, when they're giving saline vehicle control injections, they have similar amount of dopaminergic neurons in the substantia nigra pars compacta. However, after they go the treatment with the LPS injections, we see here that the wild-type regular group that does express the receptor has significantly lower numbers of dopaminergic neurons compared to the mice that lack the receptor. And when we quantitated this, the wild-type group actually lost approximately 44% of their dopaminergic neurons in the substantia nigra pars compacta compared to only 10% in the group that lacks the receptor. So the answer to our question was that this data suggests that taking away the receptor in mice has a protective effect. Now we want to know about the ligand. So we asked, if we apply the ligand IL-13 to cells that are grown in plates, so they're cultured in plates, what effect does that have, a protective effect or a detrimental effect? And here are the results. We can start here on the left with this graph that uses, we use first mouse dopaminergic cell line. And so what we see here in blue is by itself, when we apply increasing concentrations of IL-13, it did not increase the killing of this mouse dopaminergic cell line. However, in the presence here in the red line of a sublethal dose of hydrogen peroxide, when you apply IL-13 as well, it increased the killing of these cells. Thus, IL-13 potentiated the effects of mild oxidative stress. And so a common question we get is, this is in a mouse dopaminergic cell line. What about, does this translate into humans, into human cell line? Well, indeed, we did just that. Here in the middle, we have data from a human dopaminergic cell line, the SHSY5Y cell line. And as you can see, we got similar results. And so it's a dopaminergic cell line. It's not truly a dopamine neuron. But so we went even further to validate this. And what we did is we got induced pluripotent stem cells and differentiated them into dopamine neurons. So they're derived dopamine neurons. And as you can see, we got similar results where by itself here in blue, IL-13 does not increase the killing of dopamine neurons, but then in red, in the presence of a sublethal dose of hydrogen peroxide, it potentiated the effects of mild oxidative stress. And so another question we asked are if there exist mutations in the human IL-13 system that are associated with Parkinson's disease. And for that, what we did is we got access to DNA sequence data from the Parkinson's Progression Markers Initiative. This study is sponsored by the Michael J. Fox Foundation. And so it's DNA sequences from Parkinson's disease patients and healthy controls. And so what I did initially is I screened for missense single nucleotide polymorphisms. So a SNP is basically one person could have in their DNA sequence TTAT, and another person could have TTAC. So it's a single base mutation. And so after screening the DNA sequences of these patients and controls, I found several SNPs here summarized here, and here's a data table summarizing that. However, for the purpose of this talk, I want to focus on an IL-13 SNP. And this SNP is a change from induces a change from a thymine to a cytosine. We have found it in 2 Parkinson's disease patients, and we have found that this SNP is 32.5 times more frequent in the Parkinson's disease patient population compared to the general population. And both of these Parkinson's disease patients that carry this SNP were males, and both of them were heterozygous for the SNP. And what's interesting is that these patients were 39 and 40 years old. This indicates early onset Parkinson's disease. And so what we see is that, remember, this IL-13 SNP is in the coding region of the gene. And so then what you get is an amino acid change from a leucine to a proline. And as you can see, this amine ring is now constricted. The amine is now constricted to a ring structure, which suggests that the biological function of the protein is probably altered. And the data that I showed you in regards to the cytotoxicity assays suggests this as well. And so overall, what I want to do is determine the phenotype of a rare SNP in the human IL-13 gene that we have found in individuals with early onset PD. The hypothesis is that the rare IL-13 SNP will confer a gain of function, and in doing so, increase the susceptibility to Parkinson's disease. So how have we addressed this hypothesis? Well, first, we've conducted, again, in vitro experiments using cells. And so what we did initially is we generated the proteins of IL-13, the wild-type regular recombinant human IL-13 protein, and the mutated version of that protein. Then we used those proteins to address this question. Is mutant recombinant human IL-13 more toxic to dopamine neurons? So we repeated that cytotoxicity assay that you just saw using these 2 proteins. And what we see here at the concentrations of 1 and 2.5 nanograms per mil, indeed, the mutated version of human IL-13 was more cytotoxic, significantly more cytotoxic compared to the wild-type version. And so we're also conducting in vivo studies in mice to determine the biological activity of the mouse homologue of the IL-13 variant. So what we did is we used CRISPR-Cas9 technology to generate mice that carry the SNP. So we now have IL-13 mutated mice. And what I'm doing is I'm employing the LPS injection model that induces neurodegeneration in the substantia nigra. And then I want to compare the non-mutated mice to the IL-13 mutated mice. Do the mutated mice have significantly lower numbers of dopaminergic neurons? Do they have significantly behavioral symptoms that are Parkinsonian-like compared to the wild-type mice? And biochemically, I want to assess the brain for dopamine levels as well as the pro-inflammatory cytokines between these 2 groups of mice. And the expected results, as I've said, we expect that the IL-13 SNP will confer a gain of function, which will ultimately have the ability to cause more neuronal damage. And this is science, so there's no guarantee that the mouse model will be a bona fide model of the human IL-13 SNP. And also, an alternative thing we can do if we don't see a difference in dopaminergic neuronal numbers between the two mice in that Parkinson's disease model is use another PD model, which is the alpha-synuclein AAV2 model of PD, and perhaps in a different model, we will see a difference. And so with that, I'd like to thank my boss, Bruno, my PI, Bruno Conti, the Conti Lab members, our collaborators, as well as the translational grant that supports me, and also the NSPIRE program directors and the committee members. Thank you for this great opportunity, as well as the TSRI graduate office for their continuous support. And with that, I'll take questions, and thank you for listening.